Hello, everybody. Thank you for joining us today for this session of the Susan Samueli Integrative Health Institute's research webinar series. If you don't know me yet, my name is Rick Harris, and I'm an endowed chair in the Susan Samueli Integrative Health Institute and School of Medicine here at UC Irvine. I'm also a professor in the School of Medicine in the Department of Anesthesiology and Perioperative Care. Before we get into our talk today, I want to remind you of a few Zoom housekeeping items. First of all, this session is being recorded, and we will send all registrants at the end of the talk today. They'll be given the recording, and we will post the link on our event webpage. And I believe the link is also going to be provided in chat if you need the link here. Everyone will please ask you to remain mute to limit distractions, and please communicate with us, including submitting any questions in the Q&A feature at the bottom of your screen. All of your questions will be addressed at the end of the presentation as time allows. The Samueli Institute is committed to whole person health research, education, and clinical care. Our interdisciplinary researchers are dedicated to exploring the science of disease prevention and treatment. We bring this series to you to share with you the promising integrative health research that is occurring throughout the Institute's pilot study grants and Samueli Scholar Awards. To date, the Institute has given 31 awards with the potential to enhance our understanding of integrative practices. For more information about the Institute, you can visit our website at sssihi.uci.edu. It's my distinct and great pleasure to introduce today's speaker, Dr. Xiangmin Xu, for his presentation. Dr. Xu's primary academic appointment is in the Department of Anatomy and Neurobiology in the School of Medicine, and he also holds joint appointments in the biomedical engineering, computer science, and microbiology and molecular genetics. He's an established investigator in the field of neuroscience and the director of the UCI Center for Neural Circuit Mapping. His research interests focus on neural circuit organization and function relevant to the neurobiology of sensory perception, learning and memory, as well as stress. With strong collaborative infrastructure, the Center for Neural Circuit Mapping that Dr. Xu has successfully assembled a highly talented collaborative group that includes some of the world's most outstanding scientists. He's secured major funding at the highest level and has addressed very many major scientific problems. His center has emerged as a hub for new technologies and resources for biomedical research at UCI and abroad. The Center for Neural Circuit Mapping provides multiple services and resources, including viral production, single-cell genomics, biostatistics, and software application and development services to multidisciplinary investigators at UCI and beyond. What's particularly most relevant for today's seminar, which I did not know this until I received Dr. Xu's bio, is that he actually trained in Chinese medicine, including acupuncture during his training in the Shanghai Medical University in the 1990s. He has a significant track record with acupuncture research, working closely with Drs. Guo and Malik and their colleagues here at UC Irvine in the Susan Samueli Integrative Health Institute. He's also an inaugural Samueli Scholar and recognized by the Samueli Integrative Health Institute for contributions to the field of integrative health. Currently, his team uses state-of-the-art molecular genetic functional imaging approaches established in the laboratories to deepen understanding of neural mechanisms underlying acupuncture's mediated effects, mostly using mouse models. Successful completion of this project will help establish a mechanistic framework to account for acupuncture-mediated therapeutic effects and will further establish the evidence base for acupuncture practices. The title of Dr. Xu's presentation today is UCI Center for Neural Circuit Mapping and Acupuncture Research. Please welcome me in introducing Dr. Xu for this prestigious presentation.
Thank you, Dr. Harris, for your very nice and kind introduction. I'm very pleased to have this opportunity to talk about our Center for Neural Circuit Mapping and relate to the acupuncture research program. And I hope my presentation today would initiate more interaction and collaborations with the Integrated Health Institute investigators. For this talk, I will first to give a brief introduction to our center, to our UCI Center for Neural Circuits Mapping, CNCM. And then in the context of the center's infrastructure, resource, and translation research program, and I will talk about our center's efforts toward establish a top national acupuncture research program. And specifically, I also will talk about our research of investigating neural circuit mechanism of acupuncture effects. So to start off, I would like to tell you a little bit about my lab research program. So my lab is interested in neurocircuitry. And we're interested in addressing the neurobiology questions related to sensor perception, learning memory, and recently our lab also been expanding into aging and Alzheimer's disease research. So as Dr. Harris introduced me, so I have joined the department in anatomy. I primary appointment in anatomy, but also I joined appointment in engineering, computer science, as well as microbiology and molecular genetics. So the lab actually has quite taking multidisciplinary approaches. So we're strong in developing new genetic tools for neurocircuit studies. And then we have a strong electrophysiology imaging technologies, particularly recently increased again into the in vivo neural ensemble imaging using a miniature microscope as well as two photon microscopes. And then we also apply a single cell multiomics to our neurocircuits, particularly is that we actually are now adopting the single cell spatial genomic tools for our circuit mapping. And then our analysis is further refined and informed by engineering and computational technologies. And I should mention that the lab has a broad range of funded projects, right? And our cortical circuits, the vision research, particularly the visual cortical plasticity research, is really, has allowed us to better understand and hopefully to better treat the childhood visual development disorders such as amblyopia. And there's also, I believe, in our the learning memory research would be contributed to better curing the brain disease, including the Alzheimer's disease. So our research program is not a standalone. Actually, it's part of our center program. So the Center for Neurocircuit Mapping is currently administered by the School of Medicine's research office. So we are our school medicine center. And this center is relatively new. It was established in, as you see from here, in early 2020. So our colleagues, including Dr. Sanjay Golden, Todd Holm, and Dr. Bert Semmers, and I, we developed the center plan, and it was recommended by the school medicine's research council and approved by our dean, Michael Stemmers. And this picture showing, many important people who had help to, us to establish the center. And given the increasing the multidisciplinary nature of the neurocircuit analysis, our center actually emphasized inclusion of the, engineering, computer science, and physical science faculties members on campus. In addition to, more typical biomedical researchers, in school medicine and in school of biological sciences. So, and our center, let's see, our center member, have been working collaboratively to establish transformative and translation research programs focusing our ultimate goals to develop cures for treating human disease of the nervous systems. And our membership has been growing, and currently we have 94 faculty members. We have both internal and external members. Our membership extending from UCI to external institutions, including UCSD, UCLA, University of Washington, UT Southwestern, Allen Institute, Salk Institute, and all the members are really, our memberships are connected through our shared, grants, collaborations. These are actually quite important. We allow us to have broad, collaborative network. And our research areas of focus, including neurocircuit mapping in health and disease. As listed here, Alzheimer's disease, aging Alzheimer's disease is one of the major direction. And also include the research area also including the viral genetic tool development, gene therapy, translational research of rare genetic disease. And also develop a single cell technology for spatial omics and therapeutic discovery. So I should say that the center really has been a focal point for bringing together talented basic and clinical research from different areas toward a common goal. So our initial, so this slide shows our initial success of the team grant application. And this involved our initial success involved with 3 federal grants and totaling $7.1 million. And these 3 grants, including a 4-year, including for some reason my Zoom, including a 4-year $4.8 million grants from the National Institute of Aging, a 3-year $1.8 million grant from FDA, and another $500,000 grant from the National Institute of Mental Health. And these grants support our efforts to develop a powerful new molecular tools and enhance the resources offered by our center to neuroscience researchers. So our team members are featured here. So initially myself, we have Dr. Carl Koderman, and he was a professor of neurology, and Dr. Tom Lane is director of the Center for Viral Research. He's a professor of neurobiology and behavior. And Dr. Todd Holmes, professor of physiology and biophysics. And also we have 2 physician scientists join us, including Dr. Aula Spada, professor of pathology, and Dr. Virginia Kimonis, professor of medicine. And as well as we're also joined by Dr. Liz Head in pathology and UCI Mind. So, and also we have successful in, assembling, multi-institutional teams to secure large funding, as shown up here. And this grant, we actually got $14 million from a National Institute of Health, a National Institute of Aging to study brain circuits susceptible to aging and Alzheimer's disease. As shown up here, we leverage UCI strengths, our center strengths, as well as model AD strengths. And then we also leverage strengths from other institutes, including UT Southwestern, UCSD, as well as the Allen Institute for Brain Science. And here's another example, right? And so the center now, our center now is a part of the very large NIH's Brain Initiative project. We are part of the multi-institutional, very large NIH BICAN human brain mapping project. And so this type of grant, actually, this type of grant success is very good for our team. Actually, it's very good for UCI. Right? And as you can see, that in addition, so this grant, yes, is really focused on to create human brain cell atlas, and try to map individual human brain cells, molecular features, locations, how they change over time with aging. And the team members, including Dr. Liz Head, professor of pathology, member of our center, and Dr. Adam Manuki, pathology professor and chair, and also including a few physician scientists, including Bill Young, Dr. Rob Edwards, and as well as Dr. Hannah Park and Craig Stacks. Again, I would like to, to note that to re-highlight that the center has really been a focal point, has been bringing together many talented investigators from different fields and through effective partnerships. And then so this and so recently, you can see again, it was the center really been expanding the collaborations. And for this grant featured here, we actually team up with investigators from University of Minnesota, including Dr. Michael Koo and Tim Ebner, and Dr. Bing Lin at UCI UCSD. And other UCI faculty members, including Dr. Gopi M, who is a professor of computer science, and Dr. Kim Green, professor of neurobiology behavior, and Dr. Grant McGregor, professor of developmental cell biology. And very recently, and the center also team has been awarded with another important success. Is actually is very special, part of the Boone Initiative Armamento project. We awarded with 3.8 million to boost our viral production infrastructures. And this grant will help the campus, our center, to become a national center of neuroscience resources. Right. And let's see what else I have here. Okay. And this grant actually is show another expansion of our center capabilities that we have strong. Clearly, this demonstrates a strong diversity of the institutional partnership. We team up with Harvard and the board team, and also we collaborate with minority service institutions such as Morehouse School of Medicine in Atlanta, Georgia, and Western University of Health Science in Pomona, California. And then the center, really the center, you can already see that the center members really enjoy this kind of shared success. And actually, he would like to also highlight, here, Dr. Autumn Ivy, and who is a physician scientist as assistant professor of pediatrics. And also would like to highlight Dr. Okida Kionia. She is assistant professor in microbiology and molecular genetics, and she contributed her critical viral expertise to this grant application. So leveraging the federal funding and also our local, particularly school medicine support, we have established our infrastructure and critical resources for biomedical research at the UCI and beyond. And if you go to our website, and maybe if you under the resource service, we actually have we have extensive resource and service to offer. And for today, I think I would like to highlight just a couple of the facilities, and particularly is one of the facilities, viral production facility. And so this is important because it's like viral with the infrastructure such as viral production facilities, our centers has quickly emerged as a hub for new technology development and resource generation. So viral production facilities, so far it's been up and running pretty well, and we support the UCI basic and clinical investigators using viral genetic tools, and serve the UCI research communities by custom making the genetically modified viruses. So in addition to neuroscience regions, and part of the project now is evolved to develop new therapeutics. So the recent direction we've been the single cell genomic and epigenomic analysis of the brain circuits is one of the more recent directions for the center. So because we believe that by combining the brain, I cannot see my slides. Yeah, so by combining the power of the brain mapping technology, we anticipate by bringing the single cell genomics epigenomic tools, we are have a much significant potentials. Actually, it's much. Yeah. So anyhow, so we anticipated this will have a much high impact for our research program, particularly allow us to reveal the disease modulated connectome mappers and the molecular candidate that's confer the vulnerabilities versus disease resistance in specific cell type, particularly in the neurodegenerative disease such as Alzheimer's disease. And so these new directions will benefit from our ongoing multiple funded collaborations with Dr. Bingling at the UCSD and the Model AD consortiums at the UCI. So if you so recently we published one review article. So this is to report the conceptual advances as well as the novel technology. So you may like to check out about this new direction. So the center, aligned with this direction, the center has now has strong single cell and spatial multi-omics resources, right? And so as shown here, you see the center actually now already acquired 2 Visaging Merskov instruments which allow us to do the single cell spatial transdornics. And so this is our instrumentation, and so we also will work out the data acquisition and analysis plan as you shown here. These actually is from the human brain sections, and so it's a different color indicates the different cell types. So you will be able to profile, in this case 300 gene profile. You can profiles hundreds thousands of the brain cells in situ in the brain section. So this type of technology, the Morphish technology, really transforms the way we look at the biology and the disease. So in summary, relatively briefly, I told you that with our strong collaborative infrastructure, we have successfully assembled a highly talented collaborative group, including both basic and physician investigators. And we have secured major fundings at the very highest level to take on big scientific problems. And the center has been emerging as a hub for new technology and new resource generations for biomedical research at UCI. As Dr. Harris also introduced, we also offer multiple services and resources for our local and local investigators also outside the UCI as well. So our center infrastructure and research help to position our team toward establish a top national acupuncture research program at the UCI. So in the next, I'm going to tell you some of our research investigating the neural mechanisms of acupuncture effects. So the significance of our research, I think it's anyhow. This slide is I think it's self-evident, right? So because acupuncture is part of the Asian practice of traditional Chinese medicine, a study has shown that acupuncture is effective for a variety of medical conditions. However, it is less clear how acupuncture works mechanistically. And build on the strong premise of the literature and our previous studies, we propose to use state-of-the-art molecular genetic function imaging approach established at our center to address neurosurge mechanism underlying acupuncture-mediated therapeutic effects using animal models. So as there's earlier articles published by the UCI Brain. Dr. Shyster addressed, said that integrated health is a real whole person care that's informed by evidence. So that's why it's important. Our acupuncture research using the animal models is quite important because we provide strong evidence about how acupuncture works. Hopefully we'll do so. And by using by adopting those new circuit mapping approaches, and this is quite important. Dr. Lingo, one of my collaborators, explained that this would be this has been a first time at that time it will be first time anyone study circuits using the advanced viral genetic tools, which will help in mapping the acupuncture actions. So we started at that time we started to investigate the neuroscience basis of the acupuncture effects and the mechanism of acupuncture modulation of cardiovascular function in the mouse model. Now, we focus on the acupoint 内观 P6. The P6 has been I would say has been well studied in animal models, and it's been implicated in the cardiovascular function modulation, particularly to the high blood pressure management. So since we proposed to do acupuncture research in animal models, right? And one thing that how we're going to identify the acupoint in animal models. So generally is like, so basically generally usually in the field is that acupuncture identified in humans, so this P6 is a molecule on our forehead here, right? And then you try to identify the anatomic structures analogous to human structures. In this case, this is mouse for limb, and you find there's analog structures and so determines this to be 6, and whether it can validate this through through its biological effects. So in this case, we anyhow, so for this set of studies, we focus on P6, the acupoint in the mouse limb, and although the P6 effects modulated, so it's been known being modulated autonomic nervous system in brainstem, particularly in the rostral ventrolateral medulla called RVLM brain region. So a lot of works were actually already been done earlier at the UCI, particularly in later Dr. John Longhurst's group. However, the brain activation or new innovation basis of P6 acupuncture modulation are less clear. So that's why when we initially started, we would like to focus on the P6 acupuncture research. And in our published work, we use new genetic labeling technology to map the 4 brain region activation by P6 acupuncture. So, I will not. This is a paper already published. I will not. It's already published a few years now. So I will not get to technical detail. Just want to mention that this is new technology. We basically use the force Cree. This is a new mouse, force Cree mouse line. And it cross with a report. In this case, we use fluorescent report to generate the red fluorescent protein. So this new approach is is much more sensitive, more efficient compared to the conventional ways immunostaining approach. And so compared to sham control, we found that acupuncture at the P6 markedly increase the C4 positive neurons in number of organ region. So we use C4 as segregators to measure the new activation. So this is so once basically after the treatment, so you will let the animal to express the genetic marker for a week, and then you sacrifice the animal and you section the brain. In this study, we section brain, so you look for activation. And so let's look at, so this is a brain section, and those are different brain regions. Those are, I say, basolateral part of amygdala circuits. So these, so if you look at basolateral, so there are some activations. But on the other hand, other regions like ectorectal, so one bone region involved with the activation is much more dense with activation. So we found, so anyhow, we now get, so we found a number of bone regions that activate activate much more by the P6 activation. Right, and so this instead of showing the example from images here, we show the summary here. Right, so this think about these whole mouse brains, and those label regions basically highlight which indicate those bone regions get significantly activated by six. So you can, as I shown here, the P6, really active neurons, so we have to broad bone network, including the nuclear accumbens, cross-jaw, caudal putamen, bad nucleus adders, and some of the amygdala nuclei as well, and then as well as the ecternal cortex. And with a number of networks, we also will be able to really look at the connection, activation, correlation, other things, right? So basically, the bottom message is the bottom line is here that the P6 acupuncture, this data support P6 acupuncture has potential to influence physiological function associated with a known autonomic and a non-autonomic nervous system through its effect on the multiple bone region. And it's also have mapped the neural circuits, long-range neural circuits connections underlying the P6 acupuncture modulation cardiovascular systems with our genetic modified viral tracer. In this case, we use retrograde, the pseudo-rabies called PIV. And those are genetic modified virus we have expressed fluorescent proteins. And these virus traces have been well characterized using our earlier bone mapping experiments. And fortunately, we have Dr. Singo Zhang, our team, because this is the type mapping project. This experiment, very hard experiment. You had to do the open heart surgery and then inject the neurotropic virus, the PIV, EGFR expressed PIV into heart, because it's retrograde trace. It's it is retrograde, and these virus can propagate multiple synapses. Right? So you will inject it into the heart, and it will going to propagate to the central nervous system, including the brainstem of RVM region. Right? So we already have, as I told you, we have very good knowledge about these viral traces, right? And so so we actually observe very exciting, interesting results. So we actually seeing those PIV labeled, EGFR labeled neurons in the IVM bone regions after 96 hours following injection viral traces into the mouse heart. So why is this important? Because this provides direct evidence of the IVM mediate P6 modulation heart function. And then we follow up with even more exciting experiment, because we would like to demonstrate the causal effect of the IVM neurons activation, its relationship with the cardiovascular modulation. How are we going to do that? Right. So this is still involved with genetic tagging, genetic labeling approaches. So we estimate the animals P6, the P6 acupuncture, and we have the false Cree mouse line. Instead of we tag with fluorescent protein, now we tag with the opsin, the channel opsin. So we have the mouse line. They express that channel opsin. So you stimulate and you tag with those activate IVM neurons with channel opsin, and then we follow up with implant optic fibers get into the bone regions, the brainstem bone region, and then we can shine light on these tag neurons to make it activate. And so our experiments show that in vivo epigenetic stimulation of P6 acupuncture activate genetic target RVM neurons has robust effect by lowering the heart rate. So one example case shown in D, right? So the baseline, you see the heart rate is relatively stable. and we turn on light, stimulate, activate those targeted IVM neurons. You can see the boss decrease of heart rate, and light off, the heart rate recover. So this really demonstrates the causal effect. So experiment relatively, still preliminary, but we'll be able to replicate in multiple tries in mice. So it's a very promising results. So we hope to continue to do more of this to complete more substantial sets of experiments. So, and then we also have started to investigate the neural circuits mechanism of acupuncture mediated pain management. We choose to study the acupuncture effects on chronic pain when it is applied to acupoint. To Xiangmin in the human is basically around the leg, side of the leg. So it's and also called ST36 in mouse model of chronic pains. ST36 is one of the another well-studied acupoint, and it has good identifiable location in mouse model, is mouse hyaline. So you put some people do the electro acupuncture, putting it between the two muscles. Some just it's identified corresponding acupoint in mouse model as well. Right. So the reason that we're interested in acupuncture is pain management, because it's particularly in the United States, the chronic it's been found out that acupuncture being very effective in managing the chronic pain, and particularly now that during the opioid pandemic crisis, I think acupuncture can be very appealing and non-addictive solutions to the pain management. So that's one of some of this. We started more recently to start also looking into acupuncture therapeutic effects in terms of pain, in the pain, using the pain model. We use two mouse models. This is important because the animal models can only has limitations. So we would like to at least have two different models to look at different pain, to look at acupuncture effects from different complementary models. One just use CFA model. This is also, those are typical used in the field. Another one is neuropathic pain models evoked by spinal nerve ligation. So our hypothesis is that the sensory nerve stimulation at the ST acupoint project to central bone region, that ST36 acupuncture results in modulation of neural circuit activities in high order pain processing region. So this is a kind of the modulation in the high order bone regions can alleviate the chronic pain conditions evoked by either by inflammation, CFA model, or peripheral neuropathy. That's the neuropathic pain model. And so far we got very promising results, and particularly we found out the electric acupuncture EA in the acupoint ST36 can attenuate the pain hypersensitivity in the CFA mouse models. So this set of experiment has been conducted by Dr. Hai Zhang in my lab. So Hai treated the two group, basically treated the animal with CFA developed the pain model. Basically hypersensitive pain models, and then one group of control group either without acupuncture or just sham control. The other group, you receive the electroacupuncture for 20, I believe it's for 20 minutes at these kind of parameters here. Right. And if we look at the graph here, right? So so once animals receive CFA injection, they will develop the pain models, right? Pain sensation. So the black line, you can clearly see develop the pain is just shortly after the treatment of the CFA. But acupuncture, although they also have pain sensitivities following with the CF treatment, but with acupuncture treatment, their pain threshold is much higher. They basically mean that they have much less pain sensation. So I also would like to mention that actually doing acupuncture research in animal models are quite important because the charge that acupuncture often gets is that it's subject, right? Subject, but animal, animal not supposed to know how they feel, right? So the the way we the data we look getting into, we will be able to really look at getting the biological effect. Acupuncture research is quite objective, and so this is a real provide evidence base to explain how acupuncture works. So back to here, right? And so the control group is showing significant low pain threshold after CFA treatment, and the threshold recover baseline. That's the normal CFA model work, but the acupuncture stimulation at statistically significant increase pain threshold, so indicate the acupuncture effects. So this is where, and you can see that there's there are relatively large mouse samples here. This, I think, is a very exciting, robust effect. Then we would like to know the brain activities, using the brain mapping technology. In this case, high, using the transgenic mice that can express the genetic indicate in the mouse brains. Right? And so this movie showing that, but one more time, the movie was showing it didn't work. Anyhow, so basically, he used the global mapping approach to identify which brain region, right? Because I mentioned we are interested in high order brain processing regions response to acupuncture stimulation. And so without getting too much details, so so of course we know that some sensory cortex will respond to the cortex we call stimulate profile, right? But the region we identify is called retrospinal cortex, right? These are quite clear. They see clear difference in response to acupuncture research. So now, after we identify this brain region, now high can use single cell resolution in vivo 2-photon calcium images to examine those the individual neurons in the brain region, how they activate with the acupuncture stimulation at ST. So the conclusion is that ST6, there's a lot of neurons modulated by the acupuncturists. There's some clearly modulate increased activity during the acupuncture research. And without going to further details, I can show this as the results generally consistent with the mice we have now. And then okay, so these those are very important set of experiments. Hopefully allow us to provide mechanistic evidence to explain this acupuncture's effects. And in the last sections, I would like to tell you a little bit about the exciting directions we're taking now. So first one is we now implement the whole brain mapping of acupoint specific activation. And the second direction is the single cell and spatial multiomics applications to understand the molecular neural mechanism acupuncture function effects. So the whole brain mapping approaches that's now really enabled by our technology advancement. I just told you, or I already told you about the way we're able to genetic tagging, genetic capture. But now with the new system, we have a new instruments, new technology allow us to do the whole brain mappings, right? So basically, you have this we call the approach called serial 2-photon tomography. Right? It's automated block face imaging. Right? So you basically imaging and section brain, imaging section brain. So you will be able to get the 3D volume of the brain, a whole brain imaging mapping. And this is important. Why is this important? Because now you get the intact, essentially you get intact 3D volume images without, basically without any deformation. Right? And then you have 3D volume. You can align to standardize the brain temperature. You can do multiple comparisons within a uniform manner. So let me just show show one of the data sets here. So you're rotating whole brains, right? And Let me play one more time. Okay, let's see. So rotating whole brain, and then you see the coronal section. So the whole brain will have about 300 brain sections. So you really get the whole brain volume. So and this resolution, you can have at least have several or even subcellular resolution with 2-photon imaging. So why this? So here clearly you can see the why this technology would be important, right? Because now I'm going to show you this basically is we have 4 brains, right? 4 brains have different conditions: P6 SHAM, P6EA, ST-SHAM, ST36, and these 2 are top view. Grayscale, the heat map means more activated, the neurons more warm, and this sideway, right? This sideway you can look at. And clearly, you can already see that the SHAM and the stimulation is quite different, right? They're much stronger activation, and particularly this is the region light up the retrospinal cortex. That's and also shown more in retrospinal cortex. Clearly, the retrospinal cortex looks like one of the high order processing brain regions associated with acupuncture treatment. If you look here carefully, compare with EA stimulation, ST36 stimulation, you can also clearly see some differential activation. So this is with this new technology, we can address many fundamental acupuncture related questions. So Dr. Many of you probably know that Dr. Harris and Corey team has, they got one of the only one U24 to look at, topographic atlas repository acupuncture research. This is, and so one of the issues really is that we use this type of new technology really allow us to address biology basis of acupuncture, to look at the biological facts. This is very important. And then the other question very interesting is that, with this kind of high throughput, high throughput, another is really robust, precise, well-controlled experiment. We can determine, how to determine best acupuncture treatment in terms of duration, frequency. And then something, we also in the acupuncture practice, you can do sometimes we do multiple acupuncture stimulation together. How does this work, right? And it's kind of very important to test out, single acupuncture with multiple acupuncture stimulation. What would be the difference in terms of biological effects? And also the type of whole brain mapping allows us to identify new target areas for further studies. So this is relatively first direction, whole brain mapping. And in terms of second directions, we've been interested in using the single cell technologies to measure gene expression changes in response to acupuncture. And this is important. This allows extending from the activation mapping, function imaging to gene expression mapping. And gene expression is important because the data, the experiment will allow us to examine the underlying mechanism of potential long-term acupuncture effects at the genome scale. And these some things are particularly interesting because we all know that acupuncture is not acute. Actually, sometimes the effects can last for weeks, even more than that. And what would be underlying molecular mechanism? That's I consider is one of the fundamental questions. Yeah. And so Dr. Lei Chi-Tong is leading these efforts, and Dr. Tong has already been successful in getting the single cell RNA sequencing data from the brainstem. In response from those mice treated with acupuncture, and to extend to single cell analysis, also apply Mo-Fish base. So this allows us to do the single cell and spatial biological mapping of the gene expressions in multiple brain regions, right? So for them, this is nice thing that because the spatial mappings where you can see the surface contact. In this case, you actually see a lot of brain regions here. And so here I see those are built. So in this, we can use the hundreds of gene probes to profile the molecular expressions, right? And Dr. Tan and your lab is really leading these directions. So, and so far we've been, so I just want to show you some, show you some initial results. So that here we show up that these, the gene sets enrichment analysis, for each major cell types in brainstem based upon differential gene expressions in P6 electropuncture stimulation and controls. And so you can go to anyhow. So by summarizing, just look at those heat maps here. So really, we found some interesting, one of the very promising results. So we found in neuron and oligodendrocytes, the acupuncture-induced changes enrich in the oxidase, phosphorylation, and DNA repairs. And both of those gene modules have been known associated with synaptic plasticity. So that's something very important. As I told you, my lab has been studied the cortical plasticities. So this is something we hope to see. Basically, the acupuncture will modulate brain plasticity to achieve long-term effects. Right. So to sum up, I'll publish the ongoing and proposed study are important or will be important for determining neural and molecular mechanisms of acupuncture effects. We hope to determine acupuncture activation map of targeted neural location and molecular identity physiology of the cell types in CNS. We would like to determine the neural mechanisms of neural acupuncture modulation of cardiovascular function, and also would like to determine the neural circuit mechanism acupuncture-mediated pain management. And then lastly, I hope we will be able to contribute to the SSIHI's the mission by establishing a tool. So far, I have not achieved yet, but I think we will be able to establish a top national acupuncture research program with further support. And I do want to emphasize, the practitioners will benefit with our strong research program that aim to understand the mechanistic or operational principle of acupuncture. I would like to acknowledge the funding particular funding received for the acupuncture research. So SSIHI has been kind to us. We received a 2021 Pilot Award. And also I received an inaugural Samueli Score Award. And then also we received a small local award from the UCI Bring Pilot Award. That's actually really Zhiling is leading. Zhiling is the leader for that project. And then I also I really want to thank the people in the lab and thank their wonderful people. And I already highlighted a few people through my talk. And also would like to acknowledge my collaborators. I have very broad collaborating network, and also like to acknowledge the very substantial generous support from federal agencies, including NIH and FDA. Thank you, and I will be very pleased to take your questions.
Thank you so much, Dr. Xu, for that very interesting talk with a lot of data in there and a lot of funding that you have received for your center and institute. So that's really great. At this time, I'm going to entertain some questions that the audience might have. There's some questions that are already present, and I'm going to list a couple of them, and I'm wondering if you might be able to answer them. So the first question is coming from Dr. Stephanie Chin Lui. And she's asking, and she says, thank you for the informative talk. And she's asking, how did the pericardium 6 stimulation influence the retrograde tracing from the heart to the RVLM? So she's asking, how did the neural stimulation of PC6 influence the retrograde tracing from the heart to the RVLM?
Okay, so let me take this question. So first I try to understand. So basically, I think Stephanie's question is say how. So that's the experiment I shown is use the genetic modified the pseudoretrovirus is a retrograde tracing. So when you actually injected the retrovirus into mouse heart, and with time it will propagate propagate from heart, and then getting transmitted to the central nervous system, including the brainstem region, RV, So, so that experiment we just want to map the underlying connection, the underlying connection basis. Right? So yes, the question is very good. How do the acupuncture, the acupuncture stimulation will affect these? So I think it would be something we can address later on with the physiology mechanism, because so far we only demonstrate there's connection, there's highway connections here. That's very important, right? And then we'll address how does the acupuncture stimulation affect the speed of the highway or another thing. Right. Very good question. Thank you.
Thank you. The next question is from Professor Kalpana Gupta, and she's asking a question specifically about gallbladder 30 and what its difference might be with stomach 36. I don't know if you're familiar with the gallbladder 30 point. It's like on the hip area. Okay. So she's saying, we used gallbladder 30 in the mouse model of CFA, transgenic breast cancer mice, and sickle cell disease mice with chronic pain. And they observed some variability in the response in both chronic disease models compared to the CFA model. So the chronic pain model shows a bit different than the CFA. So she's saying they could see neuromodulation at the spinal level. So what's the difference between stomach 36 and gallbladder 30 in mice? And she's wondering if variability in the response can be overcome by choosing different acupoints in different individuals to achieve maximum analgesic response.
So this seems a lot of questions, not just single question. But I think Dr. Gupta does. So first, I should point out that I don't know what about GB. GB36 is acupuncture or not. First, I need to know that. But I guess
it is. Gallbladder 36 is an acupuncture point located on the hip area.
Hip areas. Okay. So I guess this question might be related more like related to see if do you see how specific the acupuncture effect, or maybe later on will be better for us to do the multiple acupuncture stimulation. But the reason I would like to for our current ongoing study, would like to focus on ST36 because this acupuncture being much more well established, particularly in mouse model, there's an easy identifiable feature to locate it at ST36. Right? And variability and the response, you always see that in terms of response variability, I think the data we got is quite robust. Right? We have a relatively large sample numbers from animal use control and treatment was 13 to 14 sample, relatively large sample. You can clearly see that the threshold, the threshold, you see that the control group with the group with acupuncture receives the pain threshold. The sensation is much reduced for the ST36 treatment group. So regarding this, I think this is a good question. We would like to Dr. Kupota and I actually collaborating on several projects. Right? Yes, Kapana will be interested in talking with you further about this. Thank you.
Thank you. Dr. Gupta is confirming that the gallbladder 30 point is between the trochanter and the lower sacral region, kind of on the gluteus kind of area of humans. I think we have time for, so there's a new question coming up by Brenda Norabe. And Brenda's saying, hello, Dr. Zhu, thank you for your incredible talk. It's exciting to see these methodologies being applied in acupuncture. Based on your research, do you think that the usage of acupuncture needles is necessary to generate the effect? Would you expect to see similar effects with simply doing acupressure?
So Brenda, I think these questions, you ask very good questions. So in China, so as Dr. Harris introduced me, I was study medicine in Shanghai Medical University in 1990. So I actually study some acupuncture. Also, I learned how to do the acupressure. So I do think there's some effects. It's the reason that acupuncture is, it's generally harmless, right? With needle, there's certain thing, there's some safety issue with needles, right? And but I the and the yeah so anyhow, if the point is that acupuncture, so really I think is acupuncture, the needle stimulation versus acupressure, the internal. That's something we can address, right, even in animal model. That's I want the question to say how in terms of duration, frequency, how you stimulate the acupuncture to determine the biological factor. That's something we can address from our basic science research. And also I should mention that you mentioned that we have used a lot of state-of-art technology. These days, there's one of the paper I cite, they actually we can use epigenetic to activate the sensor terminals without use needle. I call it opto acupuncture. This is something the lab we're trying to we're actually is going to set up trying to do the optogenetic stimulation instead of needle stimulation.
Okay. Thank you everyone for joining us today. I think we're going to have to cut it off now because we're a bit over time. I'd like to thank Dr. Xu for his very impressive presentation. And I'd like to also announce that the Center for Neural Circuit Mapping is hosting a special Cajal Club and the Allen Institute for Brain Science, Structure and Function Development of Neural Circuits Conference on Monday, August 21st through Wednesday, August 23rd of this year at the Beckman Center for National Academies of Science and Engineering. I believe we've posted the link to that and more information. So we're going to post that in the chat for you. And all of our team will email all the registrants the link to this recorded session. We hope you've had the date for your next research webinar scheduled for November 15th. That's the next one. That'll be Dr. Katrine Whiteson, and she will be presenting on microbes, their metabolites, and the impact on health. Stay tuned for more announcements in your email box. And please do check out our events on the webpage. Thank you so much. And I really appreciate Dr. Xu for giving his presentation today and the audience for those excellent questions. Thank you very much, everybody.
Thank you, Dr. Harris. We'll talk next time. Bye-bye.
Sounds good. Bye.